This video is going to cover the first chapter in the course resource, and we'll be looking at the characteristics of life and the nature of the scientific process, and then we'll consider the relationship between science and social responsibility as well. So characteristics of life, the first characteristic is life is organized. And it's organized in a hierarchy of levels, first of all from atoms and molecules to cells, tissues, organs, and organ systems. So that's the first characteristic of life, that it is organized. Then living things, they need to acquire energy and materials to carry out their activities. So for example, humans eat food that supplies molecules for energy. Living things need to reproduce. So an organism's genes carry genetic information for reproduction. Living things, they respond to stimuli. For example, they move away or toward a stimulus, and this is a part of its behavior. In addition, living things, they maintain homeostasis, which is the maintaining the internal environment of the organism and keeping it relatively constant. Also, living things, they undergo development. So an example of that would be that an acorn develops into an oak tree. And living things, they have the capacity to adapt to their environment, and adaptations, they allow survival under changing conditions. Also, living things, they undergo changes of characteristics during generations of reproduction, as natural selection then ensures the survival of individuals that are best adapted to that particular environment. And living things are said to undergo evolution then, as traits in populations and species change. So we've looked at the common characteristics of living things and how then adaptations then relate to evolutionary change. So here's some of the key terms and the key concepts that we've just covered in this particular section, and so these are nicely laid out for you in a summary. Next, we'll look at the process of science, and biology then is the study of life, living things, and like any other science, biology uses the scientific method to test hypotheses. So basically, the process begins with observations, and these lead to hypotheses, which then are either tested through experiments and further observation, and from there conclusions can be made, or perhaps conclusions can be made directly from a hypothesis. And these conclusions that are generated from experiments and observations, they lead to the development of scientific theories. So scientific theories then is actually kind of the pinnacle of the scientific process, whereby we are continuously developing theories and adding to them, and perhaps taking away some things from them as experiments will dictate based upon their results. So the first part of that then is observation, and that scientists believe that natural phenomena, natural events or facts that can be detected, can be better understood if they're observed and studied. So all of the senses then are used to make observations, all of the senses that we as humans have, and understanding is expanded by taking advantage of the knowledge and experiences of other scientists. And so the sharing of information is absolutely crucial to this process as well. So observations then can lead to a hypothesis or hypotheses, and that leads then to predictions that can be tested through experiments. So scientists use what's referred to as inductive reasoning to combine known facts into a cohesive idea. So the result of this creative thinking then is a testable hypothesis, which is a tentative explanation of a natural event that can be tested. So the hypothesis then is presented, and this is the key. This separates science from non-science, is that it must be presented as a falsifiable statement. Either results will either support or not support a hypothesis. So in order to test a hypothesis, then we create and we devise experiments for further observation. So scientists then use deductive reasoning to determine how to test a hypothesis using a logic-based statement such as, if we do such and such, then we would expect such and such to occur. And such a statement then sets out a likely result if an assumption is true or not. So the hypothesis then can be summarized in a prediction. So as an example of this, a hypothesis could state that if organisms are composed of cells, then examination of an organism under a microscope will reveal cells. So the prediction would be that microscopic studies of organisms will reveal cells. Now, this might seem pretty fundamental in our age, but before the age of microscopes, this was indeed a very valid question that really needed further development in terms of the invention of the microscope in order to be able to test this actually. So then it became an actual testable hypothesis once we had the equipment in order to be able to do that. So the design of an experiment, experiments are usually conducted to test a hypothesis. However, the success of the test depends on the experimental design. So a good experimental design, it will ensure that testing is specific and the results will be meaningful. It always includes a control, a subject that goes through all the steps of the test but is not actually exposed to the factor that's being tested. So for example, in a drug trial, the experimental group receives the drug that's being tested, and then the control group then would receive a placebo. So then after the experiments are conducted, then the data that's collected needs to be analyzed, and so the results then are referred to as the data, and which is the record of what was observed during the experiment. So data is measurable, and then it must be recorded in a way that's consistent and can be analyzed. So the statistical data is usually recorded in a table or displayed in a graph. Repeated trials of the experiment result in data that can be compared. The sample size in the experiment must be large enough then to yield reliable results. So if you only have a few subjects, then that's not going to really give you reliable results. You need large numbers of subjects. A conclusion then can be drawn from the experimental data that may or may not prove the prediction and support the hypothesis. So if the conclusion does not support the hypothesis, then another hypothesis can be suggested and tested. So that leads then to scientific theories. So all of the results from experiments on a particular area of study then leads to scientific theories. And so theories that are supported by scientific observations and repeated experiments. So once a theory then has been generally accepted, it becomes known as a scientific principle or a law. And there's a couple of examples there for you. A controlled study, we just mentioned this before, but what this means is that one or more groups will receive treatment, but another group will not receive the treatment, and that's referred to as the control group. So to ensure that the result is due to the treatment, then called the experimental variable or independent variable, all other conditions between the groups must be kept the same. So the responding variable or dependent variable then is the factor that has changed due to the treatment. So here's a simple sample kind of experiment. So a hypothesis might be that pigeon pea winter wheat crop rotation will cause an increase in winter wheat production to the same or greater extent as the use of nitrogen fertilizer. So testing out to see if crop rotation can have the same effect as nitrogen fertilizer. And so that covers this first section of the course. And so what we've looked at is to distinguish between the roles of the experimental group and control group in an experiment, and then describe the role of the experimental variable in an experiment, and the process by which scientists may test a hypothesis about an observation. And here are the key terms and concepts of the second part of this PowerPoint. And if you wanted to take some notes from that yet, this is a good summary here for you. So lastly then, science and social responsibility, the application of scientific discoveries to produce a product or develop a technique for practical purposes. This is called technology, and so we use that term quite often in today's culture. And this is what the term actually means. So many technologies are beneficial, but they may also have drawbacks. So, for example, modern agricultural practices, they helped increase crop production, but also contributed perhaps to pollution. So who in society then should decide what technologies should be used or not used, and what role do bioethical issues play in development of new technologies? These are the questions that we have to ask ourselves. We're not going to actually answer those questions, but just simply raise them for you to think about. So that's the final topic in this first video, and you can use this as a good summary and to refer back to it and ensure that you've got all of the notes that you need from it.